Howdy, this is lab report 7 and 8, biochemical tests. Oxidase test, catalase test, nitrate reductase test, urea hydrolysis test, CAMP test, coagulase test, and Kirby-Bauer susceptibility test. Group 2, Ana Martinez and
Hannah Garcia, Lab Report 6, Biochemical Tests, Fall 2024.
Introduction and significance. The purpose of these labs is to measure the enzymatic and metabolic abilities of organisms for classification, identification, differentiation of bacterial species. It also helps determine the most effective treatment options in clinical settings. The materials and methods for the oxidase test. These are the materials. Add 2 to 3 drops of oxidase reagent to filter paper. If the color is blue, discard and get a new reagent. Use a sterile cotton swab and pick a colony, then add a few drops of the reagent and give it a few seconds before observing any color changes. Background information. The test is for the test is for the purpose of identifying bacteria that contains the respiratory enzyme cytochrome oxidase. It helps distinguish the oxidase negative Enterobacteria Ca and Pseudomonas Ca, which are oxidase positive. An oxidase positive bacteria will smear will turn purple within 20 to 30 seconds, and an oxidase negative bacteria, the smear will not turn purple within 20 to 30 seconds. The materials and the methods for catalase tests. These are the materials. Pick a colony from the agar aseptically using the inoculating loop and put it on a labeled glass slide. Add a few drops of hydrogen peroxide to the culture sample on the slide. Make sure to not pick up any blood if using a blood agar medium. An alternative is to drop the hydrogen peroxide on the KIATSIA's land. Observe any bubble formation. The background information. This test determines the presence of the enzyme catalase. It is used to differentiate between genera and species differentiation. The positive test will form bubbles and the negative test will not form bubbles. The materials and methods for the nitrate reductase test. These are the materials. Aseptically inoculate a nitrate broth tube with Durham tube. For the next class time, add one dropperful of sulfonylate acid and one dropperful of alpha naphthylamine to each broth. Add a small amount of zinc, toothpick full to each broth. Observe any color changes. The background information. The purpose of this test is to determine whether the organism can reduce nitrate to nitrite or further reduce it to nitrogen gas or other nitrogenous compounds through the action of the enzyme nitrate reductase. Therefore, this test determines the presence of the enzyme nitrate reductase. A positive test will have a red color and a negative test will have clear color or unchanged. The materials and methods for the urea hydrolysis test. These are the materials. Aseptically inoculate urea agar tube by streaking it with an inoculating loop. Incubate and observe any growth and or color changes. The background information. The purpose of this test is to determine whether an organism can produce the enzyme urease, which hydrolyzes urea into ammonia and carbon dioxide. It helps identify the bacteria capable of breaking down urea as part of their metabolic process. A positive test will have a pink color and a negative test will have a yellow or unchanged color of urea agar. The materials and the methods for the CAMP test. These are the materials. Aseptically streak Staphylococcus areas at the center of the blood agar plate, a line in the middle, then streak 3 lines of Streptococcus pyogenes above the Staphylococcus area streak. Then streak another 3 lines of Streptococcus agalactiae below the Staphylococcus area streak. Do not let the streaks touch. Incubate and observe for any growth or color changes. The background information. The purpose of this test is to identify and confirm the presence of human or animal strains of group B Streptococcus agalactiae from the from other Streptococcus spp. A positive test will have a production of distinct arrowhead zone of complete hemolysis located at the point of staphylococcal streak where diffusing products of the organism, CAMO factor and beta lysine, have converged and overlapped. A negative test will not have an arrowhead. The materials and methods for the coagulase test. These are the materials, and the method is aseptically inoculate test tubes containing rabbit plasma, 0.5 milliliters, with organisms using an inoculating loop. Incubate at 37 degrees Celsius for 48 hours. Observe any changes. The background information. The purpose of this test is to determine whether a bacterium is capable of producing the enzyme coagulase, which causes blood plasma to clot. A positive test will have clot formation in the test tube after allotted time. A negative test will have no clot formation in the test tube after allotted time. The materials and methods for the Kirby-Bauer susceptibility test. These are the materials. Prepare a blanket growth of blood agars with each of the bacteria. Divide the plates with the Sharpie and place 3 different antibiotics on each space. Incubate and observe any growth. The background information. The purpose of this test is to determine the susceptibility of a bacterial strain to various antibiotics. The test helps identify which antibiotics are effective for treating infections caused by the specific bacteria tested. Check for bacterial growth or zones of inhibition. Results and observations. The following slides are the results and our observations of each of the tests performed. The pictures are our results, and the observations are based on what we recorded on our lab notebooks.
Results and observations for the oxidase test. Escherichia coli was a negative test, no color change and no presence of enzyme cytochrome oxidase. Unknown 102 was a positive test, purple color, presence of enzyme cytochrome oxidase. Pseudomonas aeruginosa was a negative test, no color change and no presence of the enzyme cytochrome oxidase. Unknown 202 was also a negative test, no color change and no presence of the enzyme cytochrome oxidase. Results and observations for the catalase test. Staphylococcus aureus was a positive test, bubble formation, presence of enzyme catalase. Streptococcus pyogenes was a negative test, no bubble formation and no presence of the enzyme catalase. Unknown 102 was also a negative test, no bubble formation and no presence of the enzyme catalase. Results and observations for the nitrate reductase test. Unknown 202 was a positive test with a red color and the presence of the enzyme nitrate reductase. Enterococcus faecalis was a negative test, which was clear with some red at the bottom but no presence of enzyme nitrate reductase. Unknown 102 was a positive test with a red color and the presence of enzyme nitrate reductase. Pseudomonas aeruginosa was also a positive test with red color and presence of enzyme nitrate reductase. Escherichia coli once again was a positive test. Red color, presence of enzyme nitrate reductase. Results and observations for the urea hydrolysis test. Escherichia coli was a negative result, with a yellow color, no presence of enzyme urease, and no production of ammonia. Unknown 202 was a positive result, with a pink color at the top and slight yellow at the bottom, indicating the presence of the enzyme urease and production of ammonia. Unknown 102 was a negative result, bright yellow color, no presence of enzyme urease, and no production of ammonia. Results and observations for the CAMP test. Streptococcus pyogenes, the top 3 streaks was a negative test. Staphylococcus aureus, the middle streak, was a negative test, and Streptococcus galactiae, the bottom 3 streaks, was a negative test. Therefore, there was no production of the CAM factor, which means it does not enhance the hemolysis caused by Staphylococcus aureus. Results and observations for the coagulase test. Staphylococcus aureus was a positive test, with clot formation in the presence of the enzyme coagulase. For Staphylococcus epidermis, it was a negative test, no clot formation and no presence of the enzyme coagulase. Results and observations for the Kirby-Bauer susceptibility test. This image was before the incubation, and this was the image of Escherichia coli after the incubation. No zone of inhibition, which means that the bacteria is resistant to the antibiotics tested as it was able to grow. For Pseudomonas aeruginosa, the zone of inhibition was 1.8 centimeters for novobiocin, which means that bacteria has immediate resistance slash sensitivity to the antibiotic novobiocin tested. No zone of inhibition in optokin and bacitracin, though, which means that bacteria is resistant. For Staphylococcus aureus, the zone of inhibition for novobiocin was 2.9 centimeters, which means that the bacteria has intermediate resistance slash sensitivity to the antibiotic novobiocin tested. However, bacteria is resistant to optokin and bacitracin as there was no zone of inhibition either. Conclusion. In regards to the CAMP test, Streptococcus agalactiae should have formed an arrowhead-shaped zone, which could be due to an error in the experiment. Errors such as incorrect inoculation, improper incubation techniques, insufficient contact between Streptococcus agalactiae and Staphylococcus aureus, different strain of Staphylococcus aureus or expired strain. Thank you.